So, dear ladies and gentlemen, my guess is that you enjoyed a sample of our Slovakian folklore, right? So, now is my pleasure to announce the first keynote lecture. And we are happy to have with us Dr. Phillip Lowe, founder, chairman, and CEO of Neurovigil. Dr. Philip Stephen Lowe made his first scientific discovery at Harvard Medical School as a teenager. While inventing new brain monitoring techniques at the University of Chicago, he was referred to the Salk Institute by Francis Crick, Nobel laureate of DNA fame, where he authored a PhD thesis on dynamic brain activity mapping, which led to his creation of Neurovigil, the neurotechnology company responsible for iBrain, a portable brain activity monitor. Dr. Lowe was appointed to dual position at the Stanford School of Medicine at the MIT Media Lab in his 20s. He chaired the first International Congress on Alzheimer and Advanced Neurotech, as well as the Francis Crick Memorial Conference. Dr. Lowe honors include the MIT TR35 Top Young Innovator Award, the DFJ Venture Challenge, and the Jacobs Ready Pioneer Award for the Global Innovation and Entrepreneurship. His inventions and discoveries have been profiled in the scientific and popular media, including Science, PNAS, CNN, Time, Newsweek, Forbes, The Economist, and The Washington Post. Dr. Phillip Lowe, the stage is yours.
Thank you very much, Lana. Thank you. I think it's good. Thank you. All right, well, thank you very much for being here. This was a very exciting performance. I was actually live streaming my brainwaves on my phone to see if it was as exciting as I thought it was, and it was. So let me start now by telling you that I'm here to actually challenge you. So you guys are toxicologists, and what I'm going to introduce today is a different way of performing a number of trials. So I'm first going to speak to you about some animal work that I did on birds. Then I'm going to speak to you about sleeping humans, then waking humans, and then a different model. So a guiding principle is obviously that we should actually look at the data and see wherever it carries us. So raise your hand if you recognize this species over here. These are Australian zebra finches, and when they sing, they produce a very repetitive pattern. What you're seeing on the screen here is frequency versus time, and the bird actually doesn't sing A, B, C, D, but when you see the same pattern, we just label it in a certain way so you can see that, in fact, there is a structure in the song. Humans do that too when they speak. And this person is actually saying it's all Greek to me, but it still has some structure. So there was a very interesting study that was published about 17 years ago. When these birds sing, obviously when they're awake, there is a very well-defined brain activity that goes on in a premotor nucleus called RA, Robustus acreastatalis. And if you just put a single dot when a neuron is firing, and you repeat these trials one after the other, when you see these vertical lines, it shows that, in fact, these birds, these neurons are acting in a very, very reliable way when the bird is singing. When the birds are sleeping, something very interesting is happening. There is spontaneous activity that goes on. So this is this sort of gray haze over here. And yet you still see these lines that are very similar to the waking activity. It appeared that these birds were rehearsing the song while they were sleeping. And this is analogous to work that has been found by Louis and Wilson at MIT, and also the McNaughton lab in mammals. So I invented a method to record brainwaves from these birds, and then I trained Sylvan Shank at the University of Chicago and Dan Margorish's lab, who actually then produced even more data. And we found some very interesting things. So this is a bird who's decided to spend the night in this food dish. And we're recording the brain waves. We're filming him with an infrared video. And his right eye is open, you're looking at him through a mirror, his left eye is closed, his right hemisphere is awake, and his left hemisphere is asleep. You're looking at brain waves here. So here's a problem. If we discover a completely new brain pattern, right, how are we going to justify that we've actually discovered something? If we use something called a supervised neural network, then it will only find things that we're trained to see. But what if, for example, one of you develops a disease that nobody's seen before? How can we train a machine to discover something that we haven't seen before? And how can we actually justify that what we find in a new species actually corresponds to what we think it does? So that led me to develop a new transform, a new type of mathematics to actually analyze brainwaves. And I worked on this for many years because this was an unsolved problem in the field. And one day I took a break. I went to the Art Institute in Chicago, and I stared at this painting here. This painting has a very important message. We take it for granted these days, but what Seurat did in about the 2.5-year period during which he painted this is something nothing short of revolutionary. He pushed Impressionism to an extreme. In other words, by just using tiny little dots, pointillism, and looking at the statistical distribution of these dots, you can actually find out that there's a lot of structure in an image. So here you see, for example, there is a monkey, there is a dog here, there is an umbrella, etc. There's still fine features that are available. And in neuroscience, too often, we have done what the Dutch masters have done, which is we look at, we try to look at every micrometer, at every microsecond, and we think that we have to understand it in detail in order to have the full picture. Maybe we could do in neuroscience something similar to what Seurat did, abstract the brain waves in such a way that the distribution of dots, and I'll show you how this is done, will give away the brain state. So, this here is a spectrogram. It is a plot that just shows us time versus frequency. And if in the show right before, if you heard very high frequencies, you would see signals, over here. And low frequencies, the signals would be over here, okay? So a violin, for example, would produce high frequency signals, but a trombone is lower frequency. And it appears from this plot that most of the information is at the very low frequencies. But it turns out that that's not true. If you normalize the data, then you notice that in fact there was hidden signal all the way here up to 100 hertz. And if we just blow up 2 hours and expand it, you can see that information is everywhere. And if at every time point we take the frequency which had the highest relative shift with respect to baseline, or the highest variability, you can see that now there is a very, very detailed structure that emerges. This is a very sensitive method. It will even pick up, for example, 60 hertz noise. But the most important thing is that it can give us filters that we can use in order to now completely create maps of brain activity. So what you're seeing over here is a 2D cross-section of a 3D manifold with every blue dot corresponding to slow wave sleep, and every red dot corresponds to REM sleep, and in cyan, in between, you have an intermediate sleep state. So every dot corresponds to 3 seconds of brain activity sampled 1 second at a time. And so this technique worked very well in all the animals in the study. It was a direct submission in the proceedings of the National Academy of Sciences, USA. And what we found out was that, in fact, we could actually have a performance that was very close to the human score, even though we were using a single channel of EEG, and even though humans will agree with one another only about 75% of the time. Here, this was birds, and the agreement rate was over 80%. And so what did this tell us scientifically? If we pool all this data from all these animals, it told us that slow wave sleep was decreasing throughout the night, that REM sleep was increasing throughout the night, that REM was elongated throughout the night, and that the space between REM sleep was decreasing throughout the night. This is textbook mammalian sleep, textbook, in birds. If we ask the algorithm to actually produce elements, to produce examples for the bird throughout the night of what the most typical slow wave sleep pattern was, REM sleep, etc., it could do this. So what you're seeing here is 3-second vignettes. On the vertical axis, you have microvolts, voltage so between 150 and minus 150 and 150 microvolts, and time is 3 seconds. So you have these beautiful delta waves on the upper left side. In the middle, upper middle, you have REM sleep. On the right side, you don't have these beautiful delta waves, and the eyes are not moving. This is the intermediate sleep state, the first one ever discovered outside of mammals. Wakefulness over here. Here, this unihemispheric sleep. We can't do this because we have a strong corpus callosum, a big superhighway between our hemispheres, and then a very odd signal this here. And I went to see the head of the sleep lab at the University of Chicago, and I said, I keep seeing these patterns. What are those? And he said, this is about 2000, 2001, he said, I did not know that you're working with rats. And I said, well, unless you think that birds are rats with feathers, I'm not. And he said, well, this is impossible because these structures need a cortex. These are K complexes. They are produced in thalamocortical loops, and we shouldn't be seeing them. So we did a double-blind study. We had one person who only scored the EEG, another person who only scored the video, and out of 102 cases, there were only 4 where you actually had a movement that occurred during the production of these signals. In other words, that appeared that these signals were endogenously produced in the brain. And this is a human K complex over here. Now, if we extract the low frequencies and the high frequencies from the same trace of EEG, you'll notice one thing. When the low frequencies are low, the high frequencies are high. Right? The low frequencies are on top, the high frequencies are on the bottom. When the low frequencies are high, the high frequencies are low. There's a pattern of interdigitation. This had been seen before in the cortex of cats, but we were seeing this now in birds without a neocortex. So it took 2.5 years to publish the study because the reviewers said, we love the techniques, we love the data, but you're telling us something that is impossible. How is it that the neocortex is not necessary and is at best sufficient for these patterns? And the editor-in-chief of PNAS stepped in and told the reviewer who was objecting, he said, if you agree with the data and if you agree with the methods, you cannot disagree with the conclusion just because you don't like it, And it was published. But it did shock some people in the field because we thought that K complexes and these up and down states are required in neocortex. And what we showed in the study is, in fact, they didn't. So I did my PhD at the Salk Institute, and the late Francis Crick, Nobel laureate of DNA fame, had recommended I work with Terry Sinofsky, a pioneer in computational neuroscience and a wonderful mentor. And before he passed, Francis was looking at my data and said, he asked a very profound question. He said, can a bird's ability to produce REM sleep tell us anything about the C word, right? Consciousness. And I thought, well, let me think about this. So I thought about it for a couple of years. And I decided that the best way to answer that question was just to look at data. And this was long after Francis had died. In fact, 5 years ago, there was a congress that I organized in his honor in Cambridge on this very question about consciousness in non-humans. Now, if we don't define consciousness, how can we actually look at consciousness in non-humans? And so what we thought about was the following thing. If we make a crude assumption that the brain is like a computer, and it's not. People used to think that the brain is like a telephone. In a few years, they'll call it a hologram. People always believe in a sort of very narcissistic way that the brain is like the latest technology. But if you make the crude assumption that it's like a computer, and if you make the crude assumption that consciousness is like a software that operates in this computer, are the non-human brains that different from us that we would have to assume that only our brain can run the consciousness program, right? So in other words, do they pass the Turing test for consciousness? And so we just decided to look at data. So this was in Cambridge, a one-day conference. And the first thing you realize, if you just look at the weight of non-human brains and the human brain, we don't have the heaviest of brains, okay? There are plenty of brains that are much heavier than ours. Do you know what these brains are? What is the brain on the right here? Speak up. Okay, very good. What is the brain on the left? Pardon? I have 2 votes for elephant. What else? Pardon? I've got a whale. Close. So this is dolphin. Technically, you know, you're okay. Left, orca, right brain. What's on the left over here? Pardon? Blue whale. So there are a lot of brains that are heavier than ours, a lot of brains that are more voluminous than ours, right? But people tell us, don't worry about this. Here's what we do. We will just divide the mass of our brain by our body mass, right? And if you do this, you know, it is similar to actually the infantilization quotient, which is to divide, you know, our brain mass by the expected mass of our brain given the body size. And if you do that, peekaboo, Homo sapiens is back on top, right? But this is not science. This is wishful thinking. And if you follow the argument, right, that tells us that actually fat people are dummies and babies are geniuses, right? And people say, no, Philip, you're just being a provocateur here. This is not to compare humans against humans, it's to compare humans against other species. So you say, okay, fine, no problem. So let's compare humans with, you know, insects and light birds. They have a higher ratio, a higher quotient than us. And then we're told, no, they don't count. Okay, well, you just told me that we're using these techniques in order to compare humans against other species. Why can't we talk about light birds, and why can't we talk about insects? And we're told we can't because of these neurons. These are spindle cells, von Economo neurons. And when you brush your teeth in the morning, and when you see your face, you have these cells in the prefrontal cortex that light up. And it is believed that it is thanks to this activity that you know that you're staring at yourself in the mirror. And this is called, you know, the mirror, the MST, the mirror self-recognition test, which dolphins pass and elephants as well. There's only one problem. Magpies recognize themselves in the mirror just fine. They don't have these neurons. So what we realized in Cambridge is that maybe in neuroscience we were in a similar field as astronomy was, but in the pre-Copernican era. You know, we had decided that the human brain was the nec plus ultra, and everything revolved around us. Just like we had decided that, you know, centuries ago, that our Earth was at the center of the universe, or at least the galaxy. It turns out we have to be a bit more humble, and we have to be a bit more data-driven. So we went to this room at the Hôtel du Vin, and Francis Crick was in attendance, and as neuroscientists, we decided to repudiate Descartes. So we had this Cartesian assumption that had hampered the field for many years, that basically, you know, only human brains could be imbued with consciousness. Descartes thought that only human brains could have a soul, and the idea was that animals just were biological machines, right? This came actually from the church. And we decided to put the pen to the paper and to completely repudiate this notion. And what we said was, neurobiologically speaking, there is no reason to assume that. The most parsimonious explanation is that consciousness is probably evolutionarily derived. It is a good thing for us to feel pain and pleasure. You cannot feel pain and pleasure unless you're conscious. Usually, stimuli that are painful lead you to go away from them, and that increases your chances of survival. Stimuli that are pleasurable lead you, unless you're on some drug, lead you to go into environments that maximize your evolutionary fitness as well. It is highly unlikely that consciousness just evolved straight for humans. So, at least neurobiologically speaking, we thought this was the most parsimonious thing to do. And so this was signed in the presence of Stephen Hawking in Cambridge. Okay, so there are all kinds of things you can do. You can go vegan. You can, this interested a lot of people. For a while, I spoke to people in different, color clothing. It was red, or it was white, or some fancy suits. But it reminded me of a question that I had when I started at the Salk Institute. I asked 3 eminent neuroscientists the same question. I said, if animals are conscious, why do we anesthetize them, right? And I had 3 very, very different answers. One gentleman said, well, we anesthetize them because we don't want them to move during surgery. I agree. We anesthetize them because we don't want to think that we're causing pain. So we do this for us. The other eminent scientists said, well, we anesthetize them because obviously they feel pain, and obviously, history will not look back at us kindly. I was surprised to hear this from him. He worked with monkeys. And then another fellow just said, we don't care about this. This is a political issue. We don't have to worry about this. But the time had come, obviously, to think about it. In Brazil, there were riots when beagles had been found to be drugged in a toxicology study, and people actually stormed that institute. And I was on a flight from Israel back to the US, and I was interviewed by a Brazilian paper, and I said, don't blame the toxicologists. Don't blame the scientists. They're doing their best with the training that they have, and if they're using public funding, and if the public wants them to do something else, then give them the tools, give them additional money, give them more grants so that they can actually develop new tools. But you cannot be upset at them for doing the work that is only available to them right now. In the province of Sao Paulo, the state of Sao Paulo, they decided to ban the use of animal testing for cosmetics. So, and also in Curitiba, there was a declaration by veterinarians which pretty much stated that animals are not objects, they're sentient beings, and accordingly they shouldn't be treated as things. If you wonder what the paw is over here, it is a federal seeing eye dog, CEDAR, who decided to sign the declaration as well. There was a declaration in Lisbon 2 years ago, which stated that for those people who do perform animal work, that they may want to actually write a business plan to explain in detail why this particular animal is necessary for this particular experiment and why this particular experiment is important. Also, they agreed that they would make the lives of the animals available for the funding agencies to see. And I think that for people who do practice animal work, that is not a very high bar. If you are a great animal researcher, and there are people who do their very best, this should not, cause fear in anybody. Okay, so question number 1, can we do animal work a little bit better? Yes, we can. In fact, I was not the person who discovered, or who was the first person. I was not the first person to have the concept of creating clouds out of EEGs. This study here is pretty interesting. No thresholds are used. We know what all the variables are, but instead of using one channel as I did, all the electrodes are in the brain, in the hippocampus, the striatum, the cortex, and the thalamus. The colors are interesting, but there's no comparison with the human scoring, and it hasn't been applied to humans. So some of you may say, well, hang on, what is WT? Isn't this weight training? No, these are rats. It's whisker twitching, but if you want to see a rat weight train, there you go. So I had these 2 fellows who worked with me, and I said, if we have this algorithm that is so powerful, maybe we can actually get brainwaves now from these rats without making a hole in their head. So why don't you just put a human electrode on the scalp of a rat and see what we get? So this is what they did, right? So the idea is, if you don't actually put a hole there, you're going to have a less stressed rat and less stressed human as well, but the question is, can we actually get meaningful data? So they used a human electrode, and these are 30-second vignettes. And over here, so top left is quiet wakefulness, top right, active wakefulness, bottom left, light anesthesia, bottom right, deep anesthesia. We run this through the algorithm. Deep blue is deep anesthesia, fuchsia light anesthesia, the rest is wakefulness. Not a single hole has been made in the rat's head, and this is all non-invasive. Okay. But so what, right? So what? Who cares? Let's say that we can you know, help far more animals than we do in our animal trials. Here are the numbers. So our industry worldwide every year kills 100 million vertebrates. And we burn $40 billion US. That is, if you count academia, industry, and government. And with all of that expenditure of capital and all of that pain that is likely caused, the likelihood that we will test a drug, a compound, in a human, do you know what it is? Anybody wants to volunteer? I have a misanthrope who thinks it's less than 1%. It might be. Last time I checked, it was less than 6%, which is still very, very, very low. Okay, so do you know this drug over here, Halcyon? Raise your hand if you do. That scares me that toxicologists would not know about this drug. Do you know about Ambien? Yes? Okay, so Halcyon was the Ambien of the late 1980s and of the early 1990s. It was the most prescribed sleeping pill in the world. It was prescribed in about 90 countries. And it was immediately banned or restricted in about 11 countries. And then the rest followed. In the UK, it was banned for psychiatric disturbances. In the US, we thought, well, we can live with some psychiatric disturbances, clearly. And we lowered the compound, the dosage, from 0.5 milligram to 1/4 milligram. And they did write on the inset the following statement: bizarre or abnormal behavior, agitation, and hallucinations may occur. Just in case you were worried, you know. So here's the list of side effects. This was an FDA-approved drug, right? But of course, if you look at the animal trials, I haven't met too many mice that feel suicidal, you know, and that are going to throw themselves off of the Empire State Building. So there are all kinds of things that we need to check here, right? On one hand, we do these tests on these animals because we think that they will be translational, you know. On that, so for example, diseases of the mind like depression and so on. But on the other hand, we don't want to agree that they likely are conscious, which is why we use them. We cannot have it both ways. But eventually, we have problem drugs such as this one. And it was a big problem. There was a fellow in Washington, D.C. who took it. He invaded one country. His son took 2. He might have had a stronger dosage. Without being facetious, my father was on it, and he actually threatened a person with a gun during one of those drug-induced outbursts. He was the first person to be fully pardoned by the Swiss Cantonal Parliament in Geneva when it was found that, in fact, he was acting under the influence of this compound. So what I'm going to propose to you today is a completely new way of doing business here. So I came here to challenge you and hopefully to inspire you as well. So we introduced this model in Monaco in 2010 at the Alzheimer's conference and at Messenger Technologies. And so let's look at the numbers. What I have to first convince you of is that there is a market pain and that the current status quo is not sustainable. So we have 2 billion people worldwide affected by a brain or central nervous system disorder, including 100 million in America. Since the last election, I think it's much higher. But in the US, every 66 seconds, a person is developing Alzheimer's or is thought to develop Alzheimer's. In California, where I live, if the population were to increase by 30%, the Alzheimer's rate would double. And by 2050, a very conservative estimate is that we will have, in the US, 13.2 million people with Alzheimer's. The latest estimates are 16 million. And without adjusting for inflation, this will cost us $1.1 trillion. So let's look at this. Let's look at Alzheimer's, let's look at schizophrenia, let's look at depression. If we multiply the number of people suffering with these diseases in the US by the cost of care, you end up with this. You end up with $100 billion of direct costs for Alzheimer's, 42 for schizophrenia, 37 for depression. If you include the indirect costs, you have the red numbers. $436 billion for Alzheimer's, 63 billion for schizophrenia, 210.5 for depression, right? This is happening today. You have a tremendous cost to the caregivers, right? 60% of people who take care of Alzheimer's patients are going to experience a combination of depression, anxiety, high blood pressure, diabetes, or compromised immune system. Half of the people satisfying the diagnostic criteria for Alzheimer's have never received the diagnosis. And half of the people satisfying the diagnostic criteria for depression have never received the diagnosis. And the ones for Alzheimer's have not even received the diagnosis for dementia. Okay. So then we also have people who are paying for unnecessary care, false positive. So about 60 to 75% of people who think they're depressed are actually paying for care they don't need. If we look at Parkinson's, with just 1 million people in the US thought to have Parkinson's, this is a $25 billion cost per year. And if you think you may have Parkinson's or there's something that's off with you, you may go through a battery of tests, right? So these over here, there's genetic testing, PET scans, MRI scans, CAT scans, a neuropsychological battery tests. And all of this, right, will only change the false positive rate from 35% at the onset to 24% after all these expensive tests. So you tell me that what we're doing is actually working. So it turns out that when we're sleeping, we still have depression, schizophrenia, or Alzheimer's. And our brain activity is much more variable during sleep than during wakefulness. In the back of our brain, we have this master switch, the brainstem, and it controls the release of neurotransmitters, which are molecules that suffuse our brain, and when we're awake, all these networks are turned on, acetylcholine, norepinephrine, dopamine, etc. When we're asleep, we have different combinations of neurotransmitters that turn on and off. What it tells us is that we have now a possibility to harness sleep and look at the brain for hours every night when its dynamic range is much greater than during wakefulness, and we should use it. So we know that sleep disorders are a major precursor of Alzheimer's. This here, raise your hand if you're familiar with this plot. Okay, nobody. So this is a hypnogram. And it is a very simple chart. It just tells us in what state of sleep or wakefulness a patient is across time, right? And this is the typical mammalian sleep that I was showing to you with the birds before, which is a slow wave sleep earlier in the night, and then REM sleep later in the night. Okay, this here on top is an Alzheimer's patient. This is data from Professor Sonia Israel at UCSD. This fellow doesn't get any deep sleep, wakes up a lot, has very little REM sleep. He happened to have apnea. If you put him on the CPAP, you'll see that you have a little bit of deep sleep, stage 3. You have just one REM episode and still a lot of awakenings. This is still completely pathological sleep. There was a very interesting study that came up a couple of years ago from the NIH. And basically, this was done in animals. This was done in trephinated mice. And so the skull, part of the skull was replaced with plexiglass. And one could actually image the brain of these sleeping mice. And one of the things that was discovered is that when these mice were sleeping, the lymphatic system was activated, and it appeared that the brain was clearing itself from debris. And I thought, when I read the study, this is interesting. Because if the amyloid hypothesis is correct, In other words, if the buildup of amyloid is a cause of Alzheimer's, and also the buildup of tau is a cause of Alzheimer's, then maybe the poor sleep means that these patients are not clearing these debris fast enough, and this contributes then to the pathology. And this actually became a standard model in Alzheimer's. There were other people who thought along the same lines. So with schizophrenia, we know that during stage 2 sleep, there's a very well-defined signal called sleep spindles that tends to thin out, and they're not as nicely grouped in the brains of schizophrenics or people who are likely to become schizophrenic. With depression, we know that people who have increased insomnia are at higher risk of having acute depression. This is a sensitive measure, but not a specific measure. You have this also for people who suffer from addiction, for example. Insomnia may push them back into addiction. But for those who are chronically depressed, they will fall into REM sleep very quickly, and they will stay in REM sleep much longer. If you REM deprive them, 80% of them will report not being depressed anymore. But if you let them sleep, they will make up for the lost REM, this REM rebound, and they'll report being depressed. So if 90% of Americans suffering from depression were correctly diagnosed, and if 80% of them were to take care of themselves, we would in the US save 330,000 human years of productivity per year. With Parkinson's, something very interesting is happening. In up to a third of patients, about 12 to 15 years before the onset of symptoms, people with Parkinson's will have REM behavior disorder, which means that while they are sleeping, in REM sleep, the muscles are not as relaxed as they should be. And the Montreal Neurological Institute believes that those people with RBD, if they were to live another 15 years, all of them would actually develop full-blown Parkinson's. Now that's very, very interesting because the latest treatments for Parkinson's, experimental treatments from Ivar Mendes in Saskatchewan and Ole Isaacson at Harvard, show that it takes about 8 years for these stem cells to actually integrate the proper networks and produce dopamine. So what you might be looking at is that it may be possible for people who've never actually experienced the symptoms of Parkinson's to be preventively cured. Okay, so let's look at human sleep. So here you've got these human brain waves. And raise your hand if you see any difference between the left and the right side. Good, we have some honest people in this room. Turns out it's very difficult with our naked eye to see that something fundamental has happened, that the brain has gone from one state to another. And were it not for the addition of the muscle tone over here, the EMG, and the eye movements, it would be very difficult by eye to notice that this person is now in REM sleep. And this is why REM sleep has been called the paradoxical state. The paradox is that it appears that you are in fact awake, but actually you're asleep. So we have 100 million people in America who suffer from a sleep disorder, if you include insomnia. Do you know how many people get a sleep test every year? 4%. And the reason is we torture them. When they come to the lab, these are people who have problems sleeping in their own bed without anything attached to them, right? So we bring them into the lab, and we're going to rub their head with an abrasive solution, and we're going to glue and tape electrodes throughout their bodies. And we do this all over the place, right? Around the eyes, on the head, all throughout the body, to find out how they would sleep without any of this junk at home. If you have a very good tech, it might take 45 minutes to prepare a patient like this. Usually it's about an hour. And of course, this is sort of the Heisenberg identity principle for biology. There's no ideal observer. The more diseased we think the person is, the more sensors we slap on that person, the more we contaminate that person's data. Right? And so this is the way it looked 10 years ago, and this is still the way it looks now. So I invented an algorithm that enables us to completely distinguish all these sleep patterns using a single channel of EEG. And so what you see on top, just like I did with the birds, but this is now humans, every dot corresponds to 30 seconds of EEG. You have 3 patients at the Max Planck Institute in Munich. The top is the manual scoring, which relies on all of these channels, and the bottom is the algorithm, which 10 years ago could already do this in less than a minute, right, using a single channel. Okay. For those of you who like these maps that I showed you before, the hypnograms, the manual scoring is in red. These are 4 different patients at the Max Planck. And the algorithm is in blue. So you can see it's pretty much superimposed, except that when there is a lot of movement, that creates artifacts on the screen, and the human usually throws his hands in the air and says, movement. The algorithm will actually remove the movement artifact and score the analog pattern. That's very important for people who suffer, for example, from epilepsy. And we've done all kinds of performance analyses. The most important thing is that we're nearly close to the theoretical limit. Humans agree with one another about 75% of the time. And that means, theoretically, you cannot agree with the humans more than 87.5% of the time. But what we can do is we can check 2 things. We can see how reliable the scoring is internally for the human and the algorithm using specialized maps that I'm going to show you. And we can also give the more experienced scorers a choice between the less experienced scorer and the algorithm, and they tend to choose the algorithm, right? If you have a digital thermometer and you've only been relying on mercury, you have to look also at how reliable the new measurement is. So this here is a 30-second snapshot. This is what the human sees, and it takes a long time to actually score all of this data, at least half an hour, sometimes much more. So you have the eyes on top, the muscles below, the brainwave below, and then the heart below. And the humans agree, as I told you, 75% of the time with one another. That means that if you sleep 8 hours, which none of us here do probably, it's still 8.5% of biological life that is completely unaccounted for. So let's do the trick that we did with the birds. So we've got the single channel VEG, we have a spectrogram, now we have this winner-take-all map. Okay. So now, instead of looking at just 30 seconds, we take these 30-second increments, and they are represented into these little dots over here, right? And we have a scan of the entire night, the brain the entire night, a single channel. Right, so we take a single EEG, collapse it into a dot, and we have this. Now I'm going to superimpose the results of the manual scoring here. There's no algorithm yet. And something revolutionary happened. Every major sleep and waking state has its own signature in the space defined by a single EEG. Now, remember, a few minutes ago I asked you if you could see the difference between left and right, and you couldn't. Can you see the difference between red and yellow? Raise your hand if you can. Good. So we have honest people who are not blind. So, and this is still based on a single EEG. So mathematically, everything was there. We just had to treat the data differently. So it works with the American data. It works on the German data. Same trick, even though they were using different equipment. It works in a 1-second resolution. And now it means that we can actually compare the humans to the algorithms. And instead of using, supervised neural networks that act a little bit like black boxes very often, even if we're to use those, we can actually see on these maps where the divergences and the convergences are. Okay, we can also go at a much higher resolution. Here we have 8 hours of sleep. We go from 0.1 to 100 hertz. Except for these movement artifacts here, you don't see anything over 20 hertz. It turns out we can actually clean up the signal, just like with the birds. Now you have slow wave sleep over here. You have these spindles that are not tightly packaged in people who are schizophrenic in stage 2. You've got gamma activity during REM, and even some higher frequencies that had not been seen before non-invasively. Okay. So what I'm telling you is it should be possible to actually get biomarkers of deep brain areas, whether they are, the thalamus, the hippocampus, or even more higher up the cortex, from just a single EEG. So we built a device called the iBrain, and this is iBrain 1. It was our first device. It's been on the market since 2009. This is my ex-fiancée, so she's also on the market. But the most important thing that I'll tell you about iBrain is the data. So this here is iBrain data. And you have about several examples, about 20 seconds or so, from the same person sleeping throughout the night. You have wakefulness here with these alpha waves. If you see these alpha waves during REM sleep, you know the person you're looking at is at high risk of having fibromyalgia. Stage 1 sleep below, you don't want to see any of this in people who are driving, for example. Stage 2 sleep, the K complexes, if they're missing, you know the person you're looking at is at higher risk of schizophrenia. Slow wave sleep, these delta waves, they tend to disappear in individuals with Alzheimer's. And finally, REM sleep, if you see too much of it, it may be a telltale sign for depression. And you even see these beautiful triangular eye movements as well. Okay, so what I'm telling you is, for those of you who are engineers, that one innovation we've done is we went from a system that is completely portable, very easy to use, that you don't have to use in a hospital, and it doesn't require any staff, and we have actually changed the transfer function. So not only are we not just looking at sleep, we're using sleep to evaluate brain fitness. And our first clients were in the pharma industry. And we get much more data as well. So we're putting less junk on people, and we're getting much more data out of them. So over the years, this is now data that we've been collecting in-house. And I'm going to show you a few examples to demonstrate the power of this kind of technology. So first, drug effects. We did a study with a pharma company. I cannot mention which one, obviously. But to make a very long story short, we found that the more they increased the compound, the more the spindles were decreasing, and then it plateaued. And we warned this company. We said, be very careful, because this is a drug for kids, and you are messing around with the spindles, and we know what it does in adults, and we're worried that you may be putting them at risk. They did not listen to us. They invested millions more in this drug, and it failed. And we had predicted this. So this is a test we did with the Navy. So the Navy borrowed an iBrain, and they said, tell us everything about this person, and by the way, this fellow may be just fine. And so when you do the hypnogram, you notice a few things. There is REM, but not a lot of REM. There is some slow wave sleep. There's some stage 2 sleep. So it's unlikely this person is a candidate for depression. It's unlikely this person has Alzheimer's type dementia. And it's unlikely this person is schizophrenic. But this is a very dumb plot, these hypnograms, because it doesn't give us much spectral resolution, right? So if we use these other plots, now on the exact same night, we see something interesting is going on. Yes, there is a little bit of REM, but it is spectrally very choppy. Do you know in which pathology this happens? PTSD, post-traumatic stress disorder. Yes, there is, stage 2 sleep, but there's an intrusion of slow wave sleep. You know where that happens? Traumatic brain injury. And by the way, the spindle count here is much bigger. That line is thicker than in the rest of our database. You know which drug causes more spindles? You're a toxicologist, you should know that. SSRIs, selective serotonin reuptake inhibitors. So what we told the Navy was, yes, we don't think that this person has dementia, depression, or schizophrenia, but we think this person is a candidate for PTSD, TBI, and it appears that even though that individual has no depression, that you've actually treated him or her with an antidepressant, and in fact an SSRI. And within 45 minutes, the US Navy came back and said you were right on all 6 counts. This was a test that we were able to do from the patient's home, and we can do this at higher resolution as well. So autism is interesting. There is a lot of talk these days about autism being on the spectrum. And I remember when I started in the sleep field, everybody said that sleep is on a continuum. So I get very, very skeptical when I hear these things. And so the beauty about this type of analysis is we use the same devices worldwide. and the same algorithms. And that gives us statistical power that other people just don't have. So we noticed that the data coming from the US and the data coming from Europe on autistic children had something in common. And in the first 4 hours of sleep, what we would find is activation in the beta range. And it didn't appear to be a seizure. And it happened in girls with autism and boys with autism. It's far too early to call this a biomarker. It's far too early. We need to record much more data. But the autism story may actually be much more refined than you think it is, and there may be some clear brain biomarkers associated with autism. So this is an interesting story. We had a person years ago who went to the emergency room and didn't feel well, right? He didn't feel well. And they just gave him, in the emergency room, an aspirin and a sleeping pill. And he asked us if we could study his brain, and we did an N of 1 study just to see what was going on, so we did an informed consent. And here's what we found out. This poor guy was waking up every time. You can see the hypnogram below. But we also had another algorithm that worked on the second-by-second resolution in humans. And in blue is the algorithm, and in green, actually a human score blind to the algorithm. And you can see they match up fairly well. So this poor guy, the maximum amount of sleep that he got uninterrupted throughout the entire night was 18 minutes. The average amount of sleep that he got per epoch was 1.4 minutes. He was dying. And when he went to the hospital, when he went to the emergency room, they just gave him a sleeping pill and an aspirin. By the time he came to us, he had already gotten into a car accident. So, you know, we are PhDs, where we cannot diagnose, but we made the data available to clinicians who then repeated the study and confirmed it, and they cured him by giving him a $10 light box to reset his circadian rhythm. And, but the key thing was to actually get the data so he could actually get the referral. So, ALS. I met Stephen Hawking my first year of college when I was 17 years old. And I didn't expect that about 15 years later I'd be sort of in charge of looking at his brain. It was sort of interesting. This is Stephen wearing the iBrain. And one of the issues that Stephen was worried about was whether he would be able to communicate if he lost the conscious control of his cheek muscle. And I said, look, we can work with you, but we have to make sure that this benefits other people with ALS as well. And you have to be involved in the project. And he agreed, and we started immediately in his kitchen. He wrote a very nice statement, you know, confirming his participation in the study. And so I sent on November 11, 2011, this gang over, to Stephen, Helena from Oxford, and Luke from Cambridge, and Johns Hopkins. And they did something very interesting to Stephen. If we can put the video. Let me do it manually. So what they did is they pretty much asked them all kinds of silly questions. They asked him if he had a pet giraffe, if he was born in Argentina, and all of these things. And the reason they did that was they wanted to see if Stephen's brain produced biomarkers of startle, right? So we wanted to see the baseline. And it turns out the baseline was just perfect. So then everybody gave up their winter holidays, and we had people from Florida, people from the Max Planck, and others who came to Neurovigil, and we ran these algorithms that I had developed during my PhD, and another algorithm I created, and something very, very exciting was going on. It's very exciting because if you look on the upper left, Stephen is resting here. If you look at the lower left, what we asked him to do was to imagine that he could move his right hand. But on the left side, these are the traditional methods. On the right side, these are the methods that I developed, and you can see that, there are pulses of high activity that are now visible when Stephen is imagining that he can move his hand. By the way, he's not very happy that I'm using an algorithm that I initially designed for bird brains to look at his brain, but we actually get data. So in green is when we give the order to imagine, and in red is when we give the order to stop. And we detect a lot of these pulses just in the middle, sometimes a little bit even before. So we redid the analysis at NASA. This is Calvin, a wonderful fellow from the UK. And it's getting much sharper. So here Stephen is opening and closing his eyes, right? And you see these peaks over here. Here he's imagining that he can squeeze his right hand. He's imagining that he can squeeze his left foot. When I met him my first year of college, he couldn't move his feet. He's imagining that he can squeeze his right foot or his left hand. So this is pretty crazy, because what you're looking at are the first neuromarkers non-invasively discovered of intent. And of course, you know, this story is not over, so we've been doing quite a bit of work on this. We introduced a couple of results in Cambridge 5 years ago, and we've been working with this gentleman, Augie Nieto. And Augie has bulbar ALS, so it started in his head, and when we met him, he would communicate with his feet, about 40 words per minute, so he was pretty quick on his feet. So let me show you a little bit about Augie.
Augie Nieto is a fitness industry entrepreneur. In 2005, he was diagnosed with ALS and soon became almost completely paralyzed. The only parts of his body he can move are his feet. He uses them to control a speech computer. Even these muscles will eventually degenerate. But he may not lose the ability to communicate. Neurotechnology pioneer Dr. Phillip is hoping to hack into Augie's mind before it's too late. Phillip and a team of engineers are building a state-of-the-art brain reader, a device they call the iBrain.
I'm wearing, the first portable brain monitor I've ever built. And what it is doing is that it is recording my brain data. It is actually sending it over to our servers, and our algorithms can quench on it.
Every time we decide to move our muscles, billions of neurons fire miniature bolts of electricity inside our brains. Phillip's electrically sensitive iBrain can detect these electrical pulses by measuring the small changes in voltage they produce on the surface of the scalp.
By seeing the activation of particular brain structures, we can tell, this person is trying to move his hands.
Phillip thought the iBrain might help patients like Augie, who, despite their paralysis, have healthy, functioning brains that can still send signals to their muscles.
ALS patients do not have problems when it comes to their own brains. The areas that his brain is using in order to control these limbs, these areas are still working perfectly fine. They're just unemployed right now. Hi, Augie. Nice to see you again.
If the iBrain can pick up Augie's mental intention to move, Phillip could create a new means for him to communicate.
You will have a prompt that will ask you to imagine to move your right hand for 4 seconds at a time. And of course, we're going to be monitoring your brainwaves very, very closely during that time and having all our algorithms crunch the data in the background as well.
The test is successful. Phillip's iBrain recognizes whenever Augie is thinking about moving his right or left hand.
Augie had a very successful first test. We will use these brain patterns in order to control a virtual hand on the screen, which will enable him to just pick these words and pick these letters and enable him to communicate. So this gives us a lot of hope and encouragement that in fact we have a system that will work for everybody and that will enable individuals to use their brain patterns in order to communicate.
The biggest fear for someone with ALS is to be not able to communicate. That is called being locked in. With the iBrain, I will never be locked in.
So that was a couple years ago. In the meantime, something happened. He spoke. So this is the first word that Augie spelled. It was in California, Corona del Mar, May 21st, 2013, and the word that he spelled was communicate. He had all kinds of options. We're not sure if he was going to call us communist, but it was actually communicate. And we actually use the system, the interface that he's most comfortable using. Instead of letting him operate with his feet, we let him operate with his mind. But we don't want to wait until people are locked in. We want to also learn their associations, what words they tend to use. And so we have another device, the iBrain 2, which is multiple channels. And here we are working with Augie, and he's actually physically moving his cheek muscles and spelling. The other system we're building for Stephen is a little bit faster. It's not this one. So we started with the letter F, and we thought, uh-oh, what is he going to tell us now? We got the F, very good, F. We got the O, you want to go down a little bit? O. What word do you think he's spelling? It's a very important word if you're paralyzed or nearly paralyzed. He made a mistake with the R. He's removing it now. You've got F, O. You got it, you got it, you got it, you got it. O. You got it. Any ideas? Yeah. Don't touch anything. There you go. Food, you got it. So, but using the iBrain 2 and using multiple channels, now it's very easy also if you just look at brain activity, if a person wants to go left or right. So you can actually activate a maze, and you don't have to wait for, for the letters to just fly by. You can just decide to go left or right and select the letters. This here behind me is with mind. So this is the iBrain 2. There are many different functions for it. It's been now requested by the Montreal Neurological Institute in Canada to do a study on 20,000 patients with Parkinson's. So, 20,000 patients to see who has RBD, brain behavioral disorder, so that we can actually screen people who are at high risk for Parkinson's. And then you might say, well, what are we going to do? You know, you guys read from the brain, you don't write to the brain. Well, one of the things I told you was, you can certainly do a stem cell study, but the other things you can do. You can also look at the effect of food. There is quite a bit of work on dairy increasing the risk of Parkinson's, including in a very recent Harvard study. So if you know that person has RBD, it may be a good thing, for example, to cut the dairy. It's been requested by the Barrow Neurological Institute in Phoenix, Arizona for stroke. And so the idea is to monitor between the left and right hemisphere, and if you see the coherence decreasing throughout the night, then that would tell us that an event has occurred somewhere. That could be a seizure, but there are other markers for that. It could be cancer. It could be a TIA, a transient ischemic attack, or it could be an aneurysm and a small stroke. So the idea is by looking at these little patterns very, very quickly, we can then get physicians to, first of all, confirm them, and secondly, take action before a catastrophic event has occurred. There is a hospital in northern Mexico that is interested in looking at glioblastomas and other things as well. I was just there a few days ago. And again, there are pharmacological interventions, but unfortunately for glioblastomas, once they're discovered, it's often far, far too late. So by seeing if there are changes early on, maybe something positive can happen. We know that, again, with the diet, there are increased risks. We know that new 5-GC, which is a sugar that is prevalent in red meat increases the risk of inflammation and cancer fivefold. This was done in a knockout study at UCSD in Ajit Varki's lab. So again, if you are at high risk of cancer, you may want to cut red meat. But this is a friend and advisor, Elon Musk, who is a small shareholder in the company. And when I was looking at Elon and wearing the iBrain 1, I thought, well, if we have this increased dynamic range, why don't we make the device even smaller? So we built the iBrain 3, this little guy here. And it's been requested in a number of interesting places. So the idea with iBrain 3 is that it will stream your data live. And we in fact filed for the patents before phones were smart, when they were dumb phones, not before my TED Talk. And the idea is that anybody with a phone will be able to beam his or her brainwaves the way I just did while the dancers were on stage. And the idea is that we'll be able to send the data back to medical centers and evaluate any type of risk. They will evaluate the risk. So I was recently at Johnson Space Center in Houston, Texas, because they want to look at the iBrain 3 to see if they could use it for astronauts on the space station and for the Mars mission, having a very small device that can tell us if individuals have slept well and are well rested before they actually operate the most expensive piece of machinery ever built by humanity would be a good thing. With seniors, we actually have a contract now with a senior care home in San Diego and the number of others that we're working on now. And the idea, again, is to find out in seniors if they have horrible sleep, because we know that this is a precursor for Alzheimer's. And again, you might say, well, who cares? What are you going to do about Alzheimer's? Well, first of all, the more data we have, the more we can actually assess the importance of the environment and certain compounds, positive influences and negative influences. For example, it's quite shocking that it's only recently that one has discovered that Benadryl, which is an anticholinergic drug, can actually is associated with an acceleration in Alzheimer's, or is associated with a higher incidence of Alzheimer's. So again, if you're a senior and you have horrible sleep, you may want to cut anticholinergics. But don't believe me, just speak to physicians. So, by putting the electrodes so close, we still actually are able to have quite a bit of data, even though traditional methods tend to fail. And even if we remove 40% of the bandwidth, and we compare what the algorithm picks up in blue here, in black, and what a human sees in blue, we're still able to look at microarousals. So when people wake up very, very quickly. So again, if you have those and you're a senior, you may want to be off of anticholinergics. So a couple of years ago, I was at the White House with President Obama when we announced the Brain Initiative. And the idea behind the Brain Initiative is to invest in the brain, and it's on a similar scale as the US did for the Human Genome Project. And when I was there, I said, this is very interesting, but you don't have a single physician and you don't have a single engineer on your panel. And maybe we should actually work with physicians and engineers and assist them and listen to what their needs are and develop technologies that are actually patient-centric. And Congress got very excited about it. There's some draft legislation that was written about this, and of course they understood that this could actually create a number of jobs. Every neurotechnology job has an impact factor of about, or multiple, about 4.3, so about 4.3 other jobs on average that are created. So as I said, Congress got behind this in order to support it, and we looked at a number of places to actually put the neurozone. We looked at the Bay Area, we looked at Boston, we looked at Wyoming actually, Vancouver, Toronto, and Montreal. And I had long said that in order to build something of this magnitude, we would have to attract people of all stripes, right? Men, women, people of all creeds, religions, backgrounds, and they have to feel welcome. And I had also said that if the new president were somebody who doesn't have the kind of worldview, that I would take this to a different country. So that's the country. So we just have been informed by McGill University that they're giving 140,000 square feet for this project to attract new companies from all around the world, including here in Slovakia, to move to Montreal and get going. So this is not going to happen overnight. It will take us years to get everything through and approved. But for those of you who are going to stay in toxicology and, of course, abide by the Declaration of Lisbon to make sure that your work is properly documented and necessary, at least you can convince yourself that it is, that's fine. For those of you who want to transition to human work, we will have a home for you in Montreal. And this is right next to an empty hospital that we're also looking at. So, and in fact, the director of the Montreal Neurological Institute and the president of McGill have written a letter to Justin Trudeau, the Prime Minister of Canada, to now request funds to build this. So we're waiting to hear back from Prime Minister Trudeau. So about 11 years ago, one of my mentors, Rusty Gage, made a very interesting prediction. And he said that within 50 years, neuroscientists, with help from other disciplines, will have a tool available that will enable them to study, consciousness at its fundamental levels, attention, perception, etc. And it will offer insights into pathologies such as schizophrenia, autism, depression, and Alzheimer's. And so I met with Rusty last year, and he said, I made a mistake when I said this. What do you think the mistake was? Exactly, 50 years. We're doing this right now. So I want to thank you for your attention today. As I said, I came here to challenge you. I wanted to avoid just complaining about the status of the field. I wanted to actually open a new line of work that may be available to you. This innovation is not done in solo. I have some wonderful people who have been working with me either as team members or investors or volunteers over the years. Every contribution counts and is appreciated. We have people who have made themselves available to try the data, to try the technology as well. And we appreciate that very, very much. And again, the field is going to change. The days of animal research are numbered. And regardless of whether or not you agree with Edison, who just put it very bluntly, that if we continue to be savages, regardless of whether you agree with that or not, the idea is, from a cost-effective measure, it is not delivering the results that we thought it would. And so we have to now migrate towards looking at people before they're sick, treating them before they're sick, as opposed to recreating a pathology in an animal and crossing our fingers and hoping that we can actually cure the animal. If chocolate were a drug that they were tested in dogs, it wouldn't be approved, right? And there would be a big loss. So let's work on humans. And with that, I'm open to questions. Thank you very much.
Thank you, Philip, for this truly amazing talk and inspiring as well. I would like to thank once more time to our keynote speaker, Philip. And if you have questions, please, Philip will be here and will be answering your questions in the foyer. I would like to invite you now to the welcome reception to enjoy the drinks, to enjoy the food. And I wish you happy Congress and enjoy the evening. Thank you.
Thank you very much.